we have studied about transcription, and after this transcription we have to study translation. That the RNA which is transcribed from DNA will enter into the process of translation. Before that, we have to discuss about the genetic code and tRNA, because their role is there in the translation process. Now, genetic code is the code which will help or direct the sequence of amino acid during protein synthesis. That's why genetic code which is helping in the translation process. Now, what is this genetic code? As we all know, there are 4 nitrogenous bases, but the number of amino acid is about 20. So, these 4 nitrogenous bases are not sufficient for coding 20 types of amino acid. For that, it was given that it may be in duplet condition, that is 4 into 4, then the number will be 16, and that is also not sufficient for 20 amino acids. Then George Gamow given the idea of the genetic code must be triplet. That means 4 into 4 into 4, and in this way it will give rise to 64 type of genetic code. Now this 64 genetic code is sufficient for 20 amino acids. That's why genetic code is of triplet type, and he has given, George Gamow was the physicist, given this idea, given this proposal that this genetic code is triplet in nature. And for proving it, so many chemical methods, so many scientists have done a lot of work, different groups have given different experiments. Like chemical method was developed by Argobind Khurana, then Marshall Nirenberg also given so many experiments, given some conclusions, and so many scientists have given, and accordingly after that, all of them have concluded that the genetic code is triplet in nature. That means one genetic code consists of 3 nitrogenous base, that is known as triplet condition. Now there are some salient features of the genetic code. Which type of genetic code exactly, how it is working, for that some salient features are there, and then the 6 important salient features are there. First is the genetic code is triplet in nature, that we have already discussed. Out of that 61 will code for amino acid. Now 1 will act as the initiator codon, and that is AUG. And when AUG is there, it have also some other function, that it will code for the amino acid that is known as methionine. Rarely, when AU is not available, then only GUG will act as the initiator codon, and the amino acid will be valine in place of methionine. Now, the second salient feature is the genetic code is specific or unambiguous. That means one genetic codon will only code for a single amino acid. That is, not any this genetic code will code for more than one amino acid. That's why it is known as unambiguous or specific. Now third point is the genetic code is degenerate. Degenerate means just opposite this unambiguous, that is some amino acid can be coded by a single codon. That means single codon will code for only single amino acid, but that same amino acid can be coded by more than one codon, and that's why it is known as degenerate. Now fourth one is codon read in mRNA in contiguous fashion. That means no punctuation is there. It will read all the all the nitrogenous bases are read, not any single punctuation or comma. That's why it is also known as commaless, without punctuation, or contiguous fashion. Now fifth one is universal. That means we will study about different types of eukaryote or prokaryote, from the bigger complex organism to the bacteria. In all the cases, the same type of amino acid will be there. That is 61 will act for will code for the amino acid. 1 will be the initiator codon, that is AUG, and 3 will be the stop codon. Stop codons are UAA or UGA or UAG. Either of these 3, anyone will stop the process of this translation, and that's why they are known as stop codon or nonsense codon. Now But some exceptions are also there, like some mitochondrial codon and some protozoa. This is not universal for that, but actually it is universal. Now, sixth point is AUG has dual role. That means it is not only acting as the initiator codon, but it is also coding for the amino acid, that is methionine. So it have two functions. First is the as initiator, and second is it will code for methionine amino acid. Now, what is the role of genetic code? If there is any mutation, then what will be effect on the phenotype or genotype? That we can understand through this example, because we are knowing that it is reading in continuous fashion, no punctuation is there. So we can see here Ram has red cap, because no punctuation will be there. That's why if suppose any, here we are inserting a single base pair, here you can see, now it will read as RAM HAS, then because B is added here, inserted here, so BRE, then DCA, then P. So you can see what will be the effect of this on the phenotype and genotype. It is totally changing the meaning. Or if we are deleting some, here we are suppose deleting H from here, H is deleted, then it will appear like that, RAM ASR EDCAP. In this way you can see, if insertion or adding or deletion, it will result into the mutation that is known as frame shift insertion or frame shift deletion. All these will result in the mutation, and this mutation may give rise to a big drastic change in the phenotype as well as genotype. Now, what is tRNA? It is also known as the adapter molecule. Its other name is soluble RNA also, sRNA, and it acts as the adapter molecule, and it has the structure of this tRNA is like that is known as clover leaf model. It has an anticodon loop. You can see here this is anticodon loop, which is complementary to the genetic code. Then this region is known as amino acid, for this end is known, which is for binding the amino acid. So two places are there, and then this tRNA is specific for each amino acid. That means for all the amino acids, single tRNA is there. It is specific for that. Or in this way, tRNA will help in the process of translation that we'll discuss through the diagram also.
In this slide we can see the genetic code. This term genetic code was given by the scientist George Gamow. We can see picture of this scientist here. He had postulated that 3-letter code must be employed to encode the 20 standard amino acid, because we know that number of that base pair in DNA is 4 only, RNA is 4 only, but the number of this amino acids is 20. Now number 4 is not sufficient for 20, and if we say that it will be duplet, then it will be 4 into 4, that is 16. That is also not sufficient for 20 amino acids. So he had given that about 3-letter code, that is genetic code is triplet, that is sufficient, that is 4 into 4 into 4, that is 64, and that is sufficient for encoding 20 types of amino acids. So the term given by him, that is George Gamow, genetic code. And actually the genetic code is a dictionary that corresponds with the sequence of nucleotide in the RNA and sequence of amino acid and protein. Now genetic code is a set of rules by which information encoded in genetic material, that is a sequence of RNA or DNA, and then it is translated into the protein by the living cells. So after the discovery of the DNA structure, then the genetic code came in existence. Here in this slide we can see the structure of this genetic code, and we can see that 3 letters are there because it is triplet. So first letter, this one, This row is showing the first letter, then this row is showing the second letter, and this one is the third letter. And in this way we are getting about 64 types of these codes. Now these, out of these 64 codes, the 3 are UAA, UAG or UGA is known as a stop codon, and it will help in the termination. And AUG is the that initiator codon, and which is coding for methionine also. So AUG has dual role, and if AUG is not available, then in rare case GUG will code for that initiate the coding process, and then the amino acid will be valine in place of methionine. Now here we can see these are the 3 stop codons. Then this is AUG is the or methionine, that this is the initiator amino acid. In this slide we can see the structure of salient features of genetic code. Here see the first this salient feature is the code is universal. That means the codons code for any amino acid in any organism. The same amino acid will be coded in any organism, either it is bacteria or human being. So that's why it is known as universal. Now non-ambiguous or unambiguous means it is the code, genetic code is specific in nature. That means each codon codes for only one amino acid, not more than one. Now commaless, that means the codons are read in 5 to 3 direction and no without punctuation, no comma is there. So 3 codons will be readed at a time. Now degenerate, this is just opposite to this point, that is here some of the amino acid can be coded by more than one codon, but not a single codon will code for more than one amino acid. That's why it is known as genetic code is degenerate. Non-overlapping means 3 successive nitrogenous bases will code for one amino acid, and then other 3 successive nitrogen bases will code for another amino acid. There is no overlapping between them. Now nonsense codon, that means 3 of these codons, that is UAA, UAG or UGA, are the codons which are not coding for any amino acid. That's why they act as the terminating or the stop codon, and they will stop the process of protein synthesis. Now linear, that is the sequence of amino acid present in polypeptide chain will correspond to the sequence of nitrogenous base present in the DNA with successive nitrogenous bases, and in this way we can say it is linear. Now triplet, that means 3 of these nitrogenous bases will come together to make a single codon, and in this way 4 into 4 into 4 is equal to 64 codons are available. Out of these 61 is coding for 20 amino acid, and these 3 are the stop codons or terminating codons. Here AUG has dual role, that is it acts as the initiator codon as well as it will code for the amino acid, that is methionine. Now here we can see that these nitrogenous bases or nucleotide will 3 will code for a single codon, codon 1, 2, 3 like that, and one codon is for one amino acid.